Hi, and welcome to this webinar, Qlucore, on multi-omics and correlation between methylation data and RNA-seq. My name is Anders Johansson. I work with sales and customer support here at Qlucore. We'll guide you through these 30 minutes. I wanted to start with a couple of words on Qlucore background. We're founded in 2007 as a collaboration from mathematical science and clinical genetics here in Lund, university town in the south of Sweden. We have grown over these years now to have customers in 20 plus countries, both in US and offices, both in US and Europe, and many large industry customers coming from primarily pharmaceuticals, but also food and beverage and cosmetics and some others. We're proud of the many leading universities using our software and the 1,000+ scientific publications so far. Today's webinar will be focused around Qlucore Omics Explorer. Here we build a fast and easy to use tool we use to analyze, visualize, and explore data, even without being a statistical expert. And I want to emphasize the visual portion of this that I hope that will translate in today's webinar. We can handle many different data types. And in today's webinar, we will focus on gene expression and DNA methylation. The data will be imported using the wizard, and we have prepared the data in a TXT file. The data background or the public data set that we will use today is the TCGA pan-cancer, pan-can cohort, which was available for download in curated format from UCSC. We will use a subset of this large cohort of samples that contain both gene expression and methylation data. We'll focus on 12 different cancer types, about 1,500 samples, 88,000 variables. The 88,000 variables are consisting then of both expression data and methylation IDs. The questions we will ask the data here will be, can we find discriminating methylation sites between tumor tissue and normal tissue? After answering this question, we will look for correlation with individual methylation sites, trying to find genes that are positively or negatively correlating with this significant methylation sites. Steps that will be demonstrated, we will do data overview and visualization, reduction of data to cover the breast invasive carcinoma that we'll focus on. We will run a statistical test to find the discriminating methylation sites between tumor and normal tissue, and then run the correlation study and generate the individual variable list of interest. Before running this import step, I have merged these two data sets. And I want to have individual control over these different data sets once imported. So I have added a new variable annotation called type here, which will indicate what type of data it is. And then we will import these things. Each sample have both RNA and methylation data associated with it. But let's skip the PowerPoint world and move to the data analysis part of this webinar. We will start totally from the beginning here in this set. So I will open using the wizard. I will go and find the data set that we're focusing on, which is the merged data set. Even though this contains 80,000 plus variables and so many samples, Qlucore can easily read it and handle it. So the first question we get, what type of data is this? This is not raw counts. The RNA has been normalized and also the methylation site. So this is other. I can also mention then, here we can see the 4 sample annotations. We have the 2 variable annotations here. So I will continue. Next, select separator. How is this organized? This is not column-based, this is matrix format. So I tell then click next. Yes, the rows, each row is one variable. Data matrix. First, we are asked to mark the top left. Do that, click next. Next question is select the bottom right cell. So I go and click the bottom right cell. Variable annotation. Here I want to click both of them. Same thing for sample annotations, but here we have 4 different sample annotations. First one, the individual sample ID, then sample type, primary disease, and last sample type ID. So we will finish this. Qlucore will import this, create a Qlucore data file. Then once imported, when there's huge data sets like this, the first plot which is shown is the table. We will move this and illustrate this as a PCA plot. We can easily do that even though there are 88,000 variables. And here I want to emphasize here, I'm not running this on any supercomputer of any kind. I'm running this on a 3-year-old standard laptop and still are able to handle these large data sets. Let's explore these annotations a little bit on the sample side. So here we have primary disease. In this case, I will choose to narrow down on the breast invasive carcinoma. Reason for choosing this one is that here on this specific disease, we have both normal tissue and tumorous tissue. Let me just show you how you can easily look at the data, even if it's this huge. So we can easily color from the individual disease areas. And we see that whatever this red one, ovarian cirrhosis, we have an outlier here, but we will neglect this finding for now and narrow down on the breast invasive carcinoma. So I will deselect all, then choose to select breast invasive carcinoma. So now we're focusing on these. Even here we have an outlier, but for the in the interest of time, let's move on and see how this is dealt with in the. And then I investigate yet another sample annotation. And here I want us to focus on the primary tumor and the solid tissue, normal solid tissue. And I want to color according to this because this is what we will focus on in this beginning part. Here I can see then that we have fewer solid tissue normal. Next question we will ask now, what are the significant differences between these two groups? And before doing so, we need to remind ourselves that we are now looking at a combination of methylation data and gene expression data. I want us to break this up into the individual, so I will use the search function here. I will search for RNA and then copy this group and create a variable list specifically for RNA. Then do the same thing for methylation. Make this correct. There. So now we have this 60,400 plus 2,700 becoming 88,000 there. We can now look at this as from the individual, either just choose to look at the RNA. Yes, they separate, but two real outliers here. Something is really different with these two. Same thing here. Let's look at only the methylation sites. And here we have one strong outlier, which is influencing these ones. That's a sensitive topic among data analysis, what to do with this. For now, in the interest of time, I will skip this and you will see this. I will now run a statistical test because I'm interested in the difference between discriminating methylation sites between this green group and the red group. So let's set up a statistical test, narrow this down to the methylation site, to primary tumor versus solid tissue normal. And when I say running the statistical test, this means that using this slider, I will now move further and further to the right. And as you can see down here to the left, I am taking away variables that don't pass the criteria we put on the corrected p-value. As we do this, we see a little bit of clustering. So there is something else going on here in this data set that we don't fully understand. There's other variation. But let's continue and focus on what we're interested in here. So I will continue to run this down into the 50 most discriminating variables to the 50. 53 is close enough. So we have generated a variable list now, which are the 53 most discriminating methylation sites between tumorous tissue and normal tissue. Let's copy this and just rename it. The 3 most discriminating sites, tumor and normal. Good enough for us for now. Keep track of this. The next thing that I want to do is that I want to populate this, what we call the column selector, and just choose the Q, which is our designation for corrected p-value, and we see that this one CG 076591914 is the most discriminating methylation island, and it's really significant. So moving forward here, yes, we have been able to run a statistical test. We have many significant variables to choose from between these two groups. Let's investigate the up and down regulation a little bit more into detail. I will choose a new synchronized plot. I will keep this plot synchronized to the previous plot and the statistical test already done. Choosing heat map, go to view, and here I will auto size it to the available window area. As for order, let's order them according to the sample type, and then the variable, let's do hierarchical clustering on them to get up and down understanding of the up and down regulation, and then a little bit of coloring on just to see the sample type. And here we can also use the color and symbol legend and the sample colors. Yeah. Here we have primary tumor are the red ones, we have the green ones. And if we look then and just use the mark tool here to show the most significant one, we see these thin white lines showing around this variable. We can draw some conclusions here. Compared to the normal tissue, in many of the samples, this methylation site is upregulated, meaning that it's more methylated than the normal situation. Let's remember that as we move forward. Before we do that, I wanted us to leave the multivariate world a little bit and focus on this methylation island and see how this behaves among the different samples using a box plot. So I again choose new synchronized plot. I move to the method and choose box sample. We will here populate the different axes. On the x-axis, I will put sample type. Here we see primary, tumor, and solid tissue. Let's choose which of the variables to visualize. And now we have visualized these two compared to each other. Yeah, we can then, before moving on, let's add medium, mean, maybe even the data points. Yes, there's good overlap, but still very significant difference between. So here now we can see significant difference in this one. One more thing that I wanted to show here, which I forgot, is that we are now looking on the normalized Z score here on the x-axis. We can change that to the original methylation values, which range from 0 to 1. And look at, so that the y-axis is changed now to this. Let's go back. And quickly then just tile the plots we have done. We started down here to the right, working in the PCA world, running the statistical test, then visualizing the result to get a feel for the up and down regulation of the methylation sites compared to the normal tissue. And then we focused in this plot here, working on the most discriminating methylation site. So moving forward from here, I wanted us to ask ourselves then, okay, we have found that this one is significantly different in the two groups. Can we find any RNAs that are correlating with this methylation site specifically? So let's investigate this. And then I will start again with a new plot. New plot means that we are starting with the original data set. No statistic test tied to this. We will go to the PCA plot. We will go back to the sample annotations and look. We are starting totally fresh, so we need to again deselect these and choose the ones that we're interested in. And choose primary solid tissue, and let's color accordingly. Now, this is not what we're focusing on now. We are looking at variables that are correlating. So I will switch to, under method, PCA, and mode, I will now go over to the variable space, because we are now going to look at correlation between the most discriminating methylation site. Just realized that I forgot to do one thing. I wanted to make a record that it was this one that I was interested. This was the most discriminating one. So I will copy that to the clipboard. I will use the search function here just to separate out this individual methylation site that I'm especially interested in. I will generate a variable list with this one specifically. So this is CG. Now that I have this, I can continue to work on this variable plot that I had in mind. But before doing that, I wanted to show how we can separate the RNA and methylation sites. I want to color them differently so we can see which one is which. So I choose yellow for the RNA. Let's do red for the methylation sites. And now we are going to look especially only on CG. This is the only methylation site that we are going to look at now. Yes, here it is. It is red. It is even marked from before. I will take that away, taking this one and moving it counterclockwise. Then we will switch to the correlation tool and mark that this is the variable that we want to correlate. Let's check the labeling also so that we're absolutely sure we have the right one. Yeah, correct. And now I want to run both positive and negative correlation study on this methylation island. So I start dragging this one further and further. See now that the first ones that appeared, went a little bit too far here. These ones over here that appeared first, these ones are positively correlating methylation island. Then we see that these dots that showed up here, they are the negatively correlating variables. There are three. We can go choose 72 here. The first methylation island shows up. Then if we go, sorry, the first gene shows up and it's negatively correlated. And we go to 71. Then we have the yes, if you choose 71.3, two genes that are negatively correlating with the methylation site that we have in mind. We have three methylation islands that are negatively correlated. I can just quickly show you, if I uncheck the negative here, we only get the positive. On the other hand, this one. I will now pursue the negative or the correlation here. We have, if we look, we have 17 variables in total that are correlating with 71.3% with this specific methylation item. Let's generate a variable list of these 7 by copying here and give this a name, 71.3% correlation with, and then it's CG 6 This variable list then contains what we're positive and negative correlation, and it contains both the RNA and the methylation sites. So here I want to just, as a side note, show you how you can separate these things. You can, of course, here shift between positive and negative and generate these variable lists individually. But as we see here that there are only 2 negatively correlated RNAs, we are going to, and no positively correlated, we are going to create a new synchronized plot and use the Venn diagram and our list tool to find these ones that are these 2 genes that are correlating with this. And we know from before then that they are, and we can also see here in the list that both of them are negatively correlated. So I would choose the list tool. I will circle this, create a new variable list. We have these 2 negatively correlated genes, and we see them below here, the ensemble IDs. So here I wanted to finish and then return back to the pile, the plots that we have worked with so far, and bring this one to the front. And again, repeat that why did I say these ones were negatively correlated? They appeared on the far side, away from the positively correlated. And I also saw that they had these minus percentage here in the list. So this is, now we need to stop and just think a little bit and what this brought us to. So I will move back to the PowerPoint world, show a little bit of, let's say, Googling work. The workflow that we have done now with data preparation and visualization, we narrowed down to the disease we were interested in. We ran a statistical analysis and then correlation study between methylation sites and gene expression. The findings then was that this CG 07651914 which is situated on chromosome 7, is the most significant methylation sites, and it shows high negative correlation with these two genes. If we look at these two genes, the first one is non-coding, situated at chromosome 7, known to have regulating capabilities. And then the other one also situated on chromosome 2, cabin 2, which when searching and investigating a little bit showed up in literature as important then for breast cancer. And so with this said, here, this is how I wanted to show how you can easily work with your data. Keep an eye out on our website for upcoming webinars. Know that you can download free trials if you're new to Qlucore. Thanks for joining. Bye-bye.